Welcome to NSTA Web Seminars, where you can find live interactive learning at your desktop. Today's seminar is part of the NGSS Core Ideas series, and it is titled Heredity, Inheritance, and Variation of Traits. Our presenter is Ravit Duncan. My name is Sue Hokinen, and I will be moderating today's program. Jeff Lehman is also online with us to provide technical support. I want to remind you to visit the NSTA Learning Center, your online portal for professional learning, and over 11,400 resources for science educators. You'll find that over 4,000 of those resources are free, and you can add them to your library to access when it's convenient for you. You can organize your resources by bundling them into collections or access thousands of other collections made by the NSTA and other teachers. Join the conversations in the community forums where you can discuss content and classroom issues with other teachers, and our online advisors are available to help you find the information you are looking for. You can also use our free tools to help you organize your own professional development to help you meet your professional development goals, and it's all available at learningcenter.nsta.org. And now I'd like to present tonight's presenters. First, I'd like to present Ted Willard. He is the Director of NGSS at NSTA with the National Science Teacher Association, and Ravit Duncan, who is an Associate Professor of Science Education at Rutgers University. Welcome, Ted. Welcome, Ravit. Ted?
Thank you. Thank you very much, Sue. Thanks, everyone, for being here. Really loving to getting involved with these explorations of the core ideas, and I think we've got one I'm really looking forward to as someone who's not really an expert in biology, to learn some myself. Want to give you all just sort of a little quick run through of some basics about the standards so we're all on the same page here. Want to start off with the idea that the standards were developed by 4 organizations. There's the National Academies, Achieve, AAAS, which is the American Association for the Advancement of Science, and my favorite, NSTA. And want to make sure we're all aware that there was a 2-step process here. First, the development of a framework, and then development of standards, which led into work on assessments, curricula, instruction, and teacher professional development that are happening based upon those standards. And so we'll start off first and take a little look at the framework itself. I'm hoping that you've had a chance to look at the framework. If you haven't, there's really no excuse not to. I highly recommend you get a hold of a copy. You can get a free copy from the National Academies Press, the address here. You can also, if you want to have a print copy, order it through the NSTA store, and if you're a member, use your discount to do so. But the main aspects of the framework are these 3 dimensions, and so what people are sometimes referring to as three-dimensional learning. And the first piece of this is the science and engineering practices, then the cross-cutting concepts, and the disciplinary core ideas. So we'll look at each of those sort of in turn. The practices are things we want students doing in the classroom as part of their learning. They're both goals and a method of achieving goals. And aspects like asking questions, developing models, analyzing and interpreting data, constructing explanations, engaging in argument from evidence. Yeah, these are the star, I would say, of the standards in the framework. Some great new work in sort of synthesizing lots of research that's gone on over the last few decades about how people best learn. Our next element here in the standards are the cross-cutting concepts: patterns, scale, systems, structure, and function. These are elements that are not unique to any one discipline, but happen to be part of physics and chemistry and biology and earth science, and fit into all of those pieces and provide some connecting tissue between them. And then finally, the disciplinary core ideas, what we're probably most familiar with: life science, physical science, earth and space science, and an NGSS engineering is also on equal footing with those other 3 discipline areas. Each piece, like the life science, breaks down into subcategories here. We're today focusing on heredity, inheritance, and variation of traits, as described here. And that piece itself breaks down into subcategories, just like all of the different topics break down further into subcategories. So all of that was described in the framework, and now we're moving on to sort of thinking about how it then fit into the standards itself. And the standards were developed by a consortium of 26 states. You can see listed in blue here, and the process was organized and managed by Achieve. So there's these 26 states, and each of these states had an important role. They had a representative, usually the state science supervisor, who was part of a committee that really made all the decisions about what happened with the standards. And then each state had to have a group of reviewers on the process. And so this was entirely a state-run effort. There was no federal involvement whatsoever in the development of these standards. What they did have then was a team of 41 writers spread across the country. There's where they each came from. And about half of those writers at the time that they were selected were actually classroom teachers, and so familiar with with students and what practices are like in the classroom. At this stage, the standards came out last spring. We have 8 states that have adopted so far: Washington, California, Kansas, Kentucky, Maryland, Delaware, Rhode Island, Vermont, and then also, really too small to see there, but the District of Columbia has also adopted. With them more on the way. The map you see here also shows you in yellow some states that have taken some sort of step in the process of adoption, whether it's been discussed at a board meeting or the state Department of Education has polled teachers about the standards and you're working, but have not yet moved to actual formal adoption. So here's an example of one of the standards, a middle school statement. We've got develop molecular models of reactants and products to support the explanation that atoms, and therefore mass, are conserved in a chemical reaction. So what we're looking at there, that is a performance expectation. And it's important to keep aware of what the role of performance expectations is. It's a combination. It's a way of blending the practice, core idea, and cross-cutting concept together to describe what should be assessed at the end of instruction. What are students able to do? It is not meant to be instructions to the teacher about what the teacher needs to do in the classroom. And so there's still a lot of freedom within the standards about what should happen here. But let's look how it pulls those different elements together. So we've got develop molecular models to support an explanation. That's the science of engineering practice of developing and using models. And then this idea of reactants and products, and that atoms and mass are conserved in a chemical reaction. That's captured here about substances reacting in characteristic ways, and that the total number in each type of atom is conserved, and thus mass does not change. Those core ideas are represented there. And then this element about mass is conserved in a chemical reaction. This idea matter is conserved because atoms are conserved in physical and chemical processes. So that's also in place here. And so all of those different elements, the cross-cutting concepts, the disciplinary core ideas, and the science and engineering practices, can be assessed through this performance expectation, the development of tasks that would do this. And so with that general piece kind of captured in here, we'll move on, let Ravit share some elements about this core idea, about heredity, inheritance, and variation of traits. Off to you, Ravit.
Thank you, Ted. I hope everybody can hear me. Thank you for the introduction, and thank you everybody for joining us, taking time away from your busy schedules to listen. I'm really excited about this presentation. Heredity is one of my very favorite ideas in the framework and the standards. And I'd like to begin by introducing myself. So I'm a learning scientist and science education researcher. I work both at the Graduate School of Education at Rutgers and the School of Environmental and Biological Sciences. And for over the past decade, I've been studying student thinking and learning in genetics. I'm in particular interested in how these ideas develop over the course of schooling. And in terms of the framework, I was fortunate enough to be one of the expert reviewers of the earlier drafts of the framework and to sort of lend my expertise, in particular to thinking about this idea, this DCI-LS3. So what I'd like to do next is try out those nifty tools that you have for polling. So we kind of did this, but those of you who joined maybe a little later, if you could mark up which grades you teach, and if you're an administrator and are responsible for multiple grades, and just, select all that apply.
Okay, and again, if you're...
I'm going to hand it over to Sue.
Yeah, thank you. If you're just joining us, the pencil tool is the third tool down. You can click on that on the whiteboard tools. You should see those on the side of the participant window now. Go ahead and click on that and then make a small mark so that everyone can be heard. There's 60 people in the room. So hopefully you guys do not have tools. Let me look. I thought I gave you whiteboard tools. You don't have whiteboard tools. Sorry, now you do. Okay, there you go. See? When you give them the tools, they can do it. Very good. And some of you found the clip art, it looks like, too. The clip art is the bottom tool. At the very bottom, if you click on it, it looks like a mountain with the sun over it. If you have access to that, go ahead and click on that, and you can put a piece of clip art on there also. And again, if you're on an iPad or if you're on some sort of mobile device that will not allow you to respond, feel free to type that information into the chat area. And then Ravit, whenever you're ready.
Yes, it seems that most of you are middle and high school with some elementary. So I'm going to try and address obviously all of the different grades, but there's probably a little more of a focus on the middle and high school, just because some of the bulk and substance of LS3 is taught at that level. Okay, so overview for today. What's the game plan? We're going to first talk about what this big idea is. Why is it such a core idea? There are actually two sub-ideas in LS3, inheritance of traits and variation of traits. Then we'll have a little pause for question and answer. And then we'll move to sort of the second part, which is looking at how students develop these ideas over time. So the progression of this idea across grade bands, and what are some of the struggles that students experience in learning genetics. We'll again pause for a little Q&A, and we'll end with a brief discussion of some teaching strategies and resources for teaching heredity, in particular for integrating this core idea with the science practices that Ted showed you earlier. And as you've seen, we're going to have a couple of poll questions along the way. So why is Heredity a DCI? Over the last 20, 30 years, there have been tremendous developments in the field of genetics. We have genetically modified food. We've cloned multiple organisms. We've sequenced even more. You can actually get a sequence of your own genes from, I think it's 23andMe. We're thinking about personalized medicine and treatments that are actually tailored to individuals' genetic makeup. So there are many ways in which, in which genetics is relevant to our everyday lives. But this idea is important even beyond the relevance to everyday life. It's really an organizing idea in biology. I really like that quote by Stephen Jones, that genetics is to biology what atomic theory is to physics. It is an organizing idea. It helps explain other ideas like evolution. If particular variations are not inherited, they're really not fodder for natural selection. This is a powerful and generative idea. It allows you to explain a variety of phenomena that may, on their surface, not look similar. And as I said before, it is relevant to everyday context. You really need to have an understanding of genetics, I think, to participate both civically and for, as an individual dealing with information about medicine, genetic screening, and so on and so forth. So let's try another little poll. Which of these is a real genetic phenomenon? And you can use your pencils to select, draw in the little bins, the little cylinders, which ideas you think are real genetic phenomena, and you can select more than one.
Okay, again, your pencil is your third tool down, or you can also use your clip art, which it seems as if some of you have found. I should add, if you're on a mobile device, you may not have access to these tools. So just go ahead and put an indication in the chat window of your choices, and you can do it simply by putting in A, B, C, or D.
Alrighty, so I'm seeing these fill up quite rapidly. And I'll give you another 5 seconds or so to put your vote down before we move on to the next slide. And here we go. So those of you that selected all of them, you're absolutely right. Genetic resistance to HIV, these are individuals that, while they're exposed to HIV, do not get sick, do not get AIDS. We have glow-in-the-dark cats, there are also glow-in-the-dark monkeys, pigs, and many other organisms. There are genetic males that look like females, and there are flies. If we fidget with particular genes, we can get flies with eyes on their legs and the rest of their body. And all of these, while they're very different from each other, all are caused by the same underlying entities and mechanisms which are at the heart of what this disciplinary core idea is about. So LS3, taken out of the framework, focuses on the flow of genetic information. The idea explains the mechanisms of genetic inheritance and describes environmental and genetic causes of gene mutation and alteration of gene expression. So that's quite a mouthful, and we're going to go over what this actually means in terms of what students are expected to know and be able to do. And as I said before, there are actually two sub-ideas here, inheritance of traits and variation of traits. So one way to think about this core idea is being comprised of 3 conceptual models. And we'll go over them one by one. The first one is inheritance patterns, right? It's this idea that we have chromosomes. Each chromosome, we have pairs of chromosomes, 23 of them. Each pair contains 2 variants of many distinct genes. And the relative dominance of variants is sort of determined by which pair you get. So you can think about it as there are multiple alleles out there. There are really multiple versions of each gene, but because you have pairs of chromosomes, you really only have 2 slots per gene. And depending on which variants you get in those 2 slots and the interactions between them, that determines, that interaction, really, the relative dominance determines the phenotype that you're going to get. So that's just patterns of correlation, right? That's a big A, little a, and then what phenotype you're going to get. And you're going to have dimples, no dimples, widow's peak, no widow's peak. But the question is that this raises, and this is sort of where Mendel was, right? These are the ideas that Mendel worked out. But then the question is, how did the variants end up in the offspring? And Mendel didn't really know a lot about this mechanism. This is essentially the meiotic model, the idea that offspring get half of the chromosomal set from each parent. And this is half a set, and that's a pretty important idea. So if you think about shoes, and you have about 4 pairs of shoes, it's not the case that you get 2 pairs, you get 4 half sets, right? So 4 shoes that don't match. And that's an important idea. We'll get back to that later. And this allows the flow of genetic information. So even though sometimes it looks like traits skip generation, it is not the case that genes sort of materialize from nowhere. Those genes are there. They may not be seen, they may not be expressed, but they're certainly there. And this is another thing that students struggle with a bit, and we'll touch upon later. This raises yet another question. Okay, fine. So you get your variants. You have your big A and little a. But how does that result in the traits, right? How does big A, little a result in dimples, for example, or in having cystic fibrosis, or a whole number of genetic phenomena? And to explain that, you need to reason within the individual. Okay, what are the mechanisms that link genes to traits within the individual? And those mechanisms are predominantly protein-based. Okay, so genes code for proteins, and those proteins have a slew of functions inside the cell, outside the cell, and essentially all the traits that we can observe are generated by proteins and their interactions and the results of their functions. So if we integrate these three models, and this is not my idea, did not come up with this. This is something from Stuart Cartier and Passmore, and it's found in a chapter from How Children Learn Science in the Classroom, which is another National Research Council publication. It's a follow-up to How People Learn, and it's really a wonderful book. I highly recommend it, and it has very specific chapters about science that are just wonderfully written. So Stuart Cartier and Passmore came up with this idea that understanding genetics really entails understanding each of these models, inheritance patterns across generations, how genes give us our traits within the individual, and how information flows from parents to offspring. And integrating them is essentially what it means to understand genetics. There's another layer, though, that's very important, emphasized quite a bit in the framework in the NGSS, and that is the role of their environment. And you can think about the environment in two ways. One aspect of the environment is the sort of the backdrop of genes that are operating together. So you think about genes, and our genome is really a network of genes that result in protein products, which can then also turn on or off other genes. So it's like little programs that can turn each other on and off. And it can get quite complex. And in fact, the reason we're having such a hard time, for example, with cancer is because of these kinds of interactions of genes turning on other genes and proteins doing all sorts of things in this sort of very complex milieu in which cancer is happening. Another way to think about the environment, though, is the external environment, okay? And there are two ways in which the external environment can affect our genes. One is through mutation. So we know, for example, that tanning is not good for you. Why? Because UV rays can actually muck up the DNA and cause breaks, actually, in the DNA molecule. There are other mutagens out there, asbestos and so on, and so forth, and that's one way in which the environment alters the gene, actually changes the recipe, okay? But the environment can also alter gene expression. So if you think about something like tanning, what happens is that exposure to UV rays causes genes to turn on, and express proteins are involved in the synthesis of melanin, which is that basically the particle that gives you that brown color, the substance that gives the skin a brown color. If you think about fruit, you know, the reason when you have bananas and they go bad really fast, to sort of ripen very fast, is because these bananas are generating a hormone, ethylene, and ethylene is sensed by the banana, and it turns on genes that result mostly in the production of enzymes that do all sorts of things like make the fruit sweeter, make the fruit softer, change the color of the peel, and give it an aroma. And so this ripening process is a result of turning genes on, and those genes are turned on because of some external factor. So the environment interacts with genes in all sorts of complex ways, and it's really critical that students understand that the environment has such an important role in determining how we look. We look the way we do because of both our genetics and the backdrop of the environment within which those genetics work. So let's do a little summary. Okay, when we're thinking about this idea, we want to think about meiosis really in terms of input and output. The idea that you have 2n and then you have half of that set. We also have sources of variation. one source of variation is sexual reproduction, right? This is in the first model that we saw. These are essentially Mendel's laws of segregation and independent assortment and recombination, which is not something Mendel knew about. We also have mutations, and we have interactions with the environment. And all of these factors result in a phenotype and works across generations. But we also have genetics working within an individual, and here the focus is on the mediating mechanisms, which is the idea that genes are coding for proteins, and proteins are a key part of this story. So I'm going to take a little break now, and I want to see what kinds of questions, if folks have questions about what I've said thus far about this DCI before we move on to thinking about how students reason about it.
So thanks, Ravit. I point out that a lot of people have loved the shoe analogy in terms of thinking of genes. It resonated with a lot of folks, and not just because I made a comment about wearing boots and stilettos together, but the hiking boots and stilettos. But Susan Zeitler's asked a question here, which I think maybe you want to address now, maybe you want to push off to later. She says, I'm sure I will get to this, but I'm struggling with how to help elementary students understand traits without overcomplicating or oversimplifying it. And maybe you have some suggestions there.
Yeah, so at the elementary grade, students are really supposed to understand that offspring sort of look similar, but not exactly to their parents. So, and I agree, I understand that traits is sort of a bit of a complicated idea because we have these acquired traits and these inherited traits, and it's a little hard for kids to differentiate. And in fact, many of those traits can't really be cleanly cut into this is genetic or this is acquired. So you might think about this: if a father has big muscles, that is acquired in the sense that the father worked up those muscles through exercise, but there might also be a predisposition for easily building up muscles that the son or daughter may also inherit. So it is actually a tricky concept in general, not just for young kids. I think that focusing on traits that kids can see in terms of color, and you can work with multiple animals or plants and look at color of flowers, that is something that little kids can really wrap their heads around. Height, various other kinds of, if you're looking at similarities and differences across species, well, dogs have 4 legs, cats have 4 legs, snakes don't really have legs, and snake offspring look like snakes in that they don't have legs, and those are pretty clearly inherited. I think it's okay, though, if kids are a little confused about that. I think it's okay if they're not sure, and I think it's okay to tell kids, well, that is a complicated trait, and it's interesting, and we're not sure exactly.
So are you? What I'm sort of hearing from you here is, and tell me if I've understood this right, is that we would like students to have some sense of what a trait is, this quality, but we don't really need to worry about whether they interpret it as being a inherited trait, a trait that's come from some interaction with the environment, or a combination of the two. Just the idea that these are these attributes that organisms have, and may notice that there's a similarity between parents and children, or between similar organisms such as dogs and cats that you mentioned there.
Yes, I think for the most part that's true. I think that getting into the weeds of exactly whether it's acquired or inherited can get tricky, and little kids do tend to have all sorts of ideas about that are not quite normative. Great.
I'm not seeing another question here. Susan Zeitler suggested an observable characteristic is basically what we mean by a trait at the early grades. I think that's what we're talking about. But I was going to say, I think we can go on now. We'll save the question time for more questions as they come up later.
Okay, wonderful. So moving on. So we're going to do a little poll. Again, we just said that understanding this gene-protein connection is really central to understanding genetic phenomena, right? And so the question is, if we're thinking about progress, well, how early should we introduce this idea? Should we introduce it in elementary school because young kids and little kids can reason about abstract ideas? Middle school because at this point they're ready to sort of reason about stuff they can't see? Or high school because really younger students don't have enough knowledge of chemistry to reason about what is essentially molecular structure. So what do you think?
Okay, and the polls are open. The poll button is the letter A underneath your name at the very top. If you hover over, mouse over that letter A, you'll see from the drop-down menu you can select A, B, or C. We only have a couple of you guys voting. I encourage all of you guys to have your voices heard. So get your votes in. And Ravit, whenever you're ready, I can lock the polls and publish these for you.
So I think once we see most people have voted, and as Sue said, vote early and often, we'll close the poll. So I don't know, maybe another couple of seconds.
Okay. I should have my voice heard also. Okay, it seems like most of you are in. If you're not able to vote for whatever reason, and you can access the chat area, then I encourage you to do that. Would you like me to go ahead and close it now?
Yes.
Okay, I'm going to go ahead and lock the responses, and I will publish this to the whiteboard so you can see the response.
All right, so we have, I would argue, a majority picking B, middle school, and some A and a little bit of C, and in fact, you are correct. So the NGSS and the NRC framework argue that middle schoolers can reason about genes as coding for proteins, and I want to make clear here that when we mean genes coding for proteins, it's this notion of genes as information for proteins, more in a recipe sort of way, and proteins as these little machines that can do things in the cell, right, that can carry out functions. We're not talking about having middle school kids understand peptide bonds or amino acids or the genetic code. That comes later in high school, and for that you do need an understanding of chemistry. But at the middle school level, we're really trying to help them reason about these protein entities, which are absolutely critical for understanding genetic phenomena. And there's been quite a bit of research that should suggest that middle schoolers can reason about proteins, especially if the protein function is a function that's familiar to them from the macro world. So if you think about a function like actin, which is a stretchy protein, or myosin, which is a stretchy protein, they can reason about something that's kind of like a rubber band or spring and what kind of properties that would have and what kinds of functions that would afford. And so that even middle schoolers can reason about an invisible protein as carrying out that kind of a function. There is this wonderful appendix, so the NGSS actually bring all sorts of appendices, all of which I think are lovely, but this one is one of my favorites. It's Appendix E, and it's the learning progression. So for each idea and each sub-idea, right, so this is LS3A, inheritance of traits, it gives you a bit of like a cheat sheet for what happens and how it develops over time. So if you look at inheritance of traits, in the early grades, the focus is really on observations and noticing that organisms, offspring, are similar to their parents, and the idea that there's some sort of inherited information that is driving these traits. Late elementary, and these are third grade to fifth grade, are also bringing in the idea of the environment as playing a role. And the environment here, you can think about, well, if you feed a dog a lot and it doesn't exercise, it's going to get fat, and some dogs kind of tend to get fatter faster than others. But how you look is not just your genetics, it's also the environment also affects the thinking about tanning, also is productive in this sense. You have a particular base color, and that varies across individuals, but then on top of that, you can also tan and become darker to some extent, depending on your base color. In middle schools, we have this focus again on proteins as affecting traits, and the idea that you can have mutations that can alter proteins and can alter the traits. And when we think about mutations here, we're really thinking about just changes to the recipe, to the information, not specific kinds of mutations. We're not talking about frame shifts and deletions here. In high school, we continue to think about it more from a molecular perspective. DNA is carrying the genetic information. Each cell in the organism has the same genetic information, but may express different genes. So if we take this recipe analogy a little further, if you have a large cookbook, it's not the case that in every kitchen we make the exact same recipes. Some kitchens may choose some recipes, and other kitchens may choose different recipes, and some may focus on desserts, some may focus on appetizers, but they all have the same book. And that's a really important idea, and it's quite counterintuitive. Students tend to think that you would only have the recipes that you need, and that's not true. And you can't actually think about stem cells and cloning if that's the perspective that you're coming from, right? Because if I can turn a skin cell back and allow it to develop into any part of the organism, well, then it must have had all those recipes in there somewhere. If we look at variation of traits, which is the second sub-idea, if you notice, and I don't know if you were actually reading or not, but the early elementary and late elementary are actually exactly the same. So it is, again, focusing on variation, inherited variation, and the role of the environment. Middle school, there's some difference here. For this particular sub-idea, we're focusing on this two-variance notion. Again, an idea that's not entirely intuitive because you only see one trait. You don't really see two traits. So it's not obvious that there are actually two alleles in there. And that you get one allele from each parent, and again, this is the notion of half a set. And that you can get variation, and variation in the middle school is explained through two mechanisms: mutations, which are changes to the recipe, and sexual reproduction, which is this segregation and independent assortment pieces, so Mendel's laws. That's it for the middle school. That's how we talk about variation. In the high school, we build on that, and we add this notion of swapping sections. So recombination here, the idea that you can swap whole sections of the chromosomes and get even more variation. And we also add this notion of environmental impact on gene expression, so more of the mechanisms of how that occurs. So if we pop up a couple of levels to think about, well, what is progressing more generally here, we can think about three ways. One is the macro-micro. So in the little kids, as I said, we're talking about traits that they can see. I think that, with third and fifth graders, you can talk about things like, well, the size of your heart and the size of your liver. They may be able to reason about having internal structures that are not visible to you. And in fact, other standards, other DCIs in life science do discuss the notion of having organs within you. So you can think about variations of those organs, but for the most part, you're staying at something that the kids can see. You can bring chicken hearts, and they can see variation in chicken hearts. Middle school and high school, we're delving into the cellular level in middle school and the proteins as these little machines. And then in the high school, we're delving into the molecular level and understanding things like the code, exactly how the code is read, idea of amino acids having particular properties that when you string them together, you get a protein with particular properties and a particular structure that affords and constrains the functions it can have. We're also deepening the causal mechanisms, right? So earlier in the earlier grades, 3rd grade, 4th grade, we're thinking of genes as information. We're not really saying exactly what the information is for. It's for traits writ large, okay? So it's information for your hair color and your height and shape of your ear. In the middle school, we're deepening this mechanism, sort of, you can think about it as opening a black box, and now we're talking about genes as instructions for protein. And then in the high school level, we're talking about, well, precisely how do these instructions work, right? How would you read the code? And overall, another way that you can think about progress is that we're adding complexity and explanatory scope. So we're adding mechanisms that allow us to explain a broader array of phenomena. Okay, so if initially you think about segregation and independent assortment, so 2 traits, let's say freckles and dimples, are separated. You can have your mom's freckles and your dad's dimples. But then sometimes there's situations in which, you know, you have traits that seem linked. They kind of always go together. And how do you explain that? You can't really explain that with Mendel's laws, and so we don't really explain that in the middle school. But you can explain that through a combination, right? The idea that, well, if 2 traits or 2 genes are sort of relatively close to each other on a chromosome, they're kind of going to go together, right? In order to separate them, you would have to break the chromosome, which is essentially what recombination does. Mutations, you deepen these ideas as well, and the environmental effects on gene expression in a more mechanistic sort of way happens at the high school level. There's another way, in addition to think about progressing, if we go back to our 3 models in the environment, is that what we're trying to do is deepen each of these models and connect them. So we're progressing by building and deepening each of these models and the connections between them rather than just necessarily repeating them. And this also suggests that at very early grades, you know, late elementary and middle, we can start talking about these models. We don't need to leave ideas like talking about proteins to the high school. So now we're going to shift gears a little bit, sort of describe, we talked about the progression and how, you know, what the research is. And those progressions, by the way, are research-based to the extent possible. It's not every single aspect of the progression has a lot of research supporting it, but for the most part they're supported, and then there's some ideas and some ages for which we have more research than other ideas in other ages, but the writers of the framework and the standards really try to base those progressions and the development of ideas as much as possible on research. So let's look at this little poll, okay? We're shifting gears to think about, well, what's hard for kids about this, right? What are some common conceptual obstacles? So try and think like a high schooler now, okay? How do you think a typical high school student would answer the following questions? And the question is, which of the following does DNA provide information for? And we explicitly tell the kids you got to choose the most accurate answer. So I'm going to tell you that too. You want to choose the most accurate answer. And I'm actually telling you what the most accurate answer is. It's C. But again, think like high schoolers. What would a high schooler say? So we're going to open the poll and give you a little time to reason about this, and then we'll see what you say.
Okay, and again, the poll buttons are located underneath your name at the very top. If you mouse over the letter A, you will see from the drop-down menu, you can select A, B, C, or D. And I am from the Chicago area, so I'm going to encourage you to vote early, vote often, and let your voice be heard. And Ravit, I can close this for you whenever you're ready. You just let me know.
Okay. Let's take a couple of more seconds. Okay, why don't we close it and see where we are?
Okay. I'm going to lock responses, and I will publish to the whiteboard.
B.
Not sure where
All
to
right.
put this. We'll
Cool.
put it
So
right
most
there.
of you think that high school students will say the traits in an individual inherits. So what I'm going to show you now is some results from a pilot that we did with high school students, okay? And these are students that learn genetics. So we did this in about May, and the kids in this particular school had genetics. If you just look at this as a regular multiple choice question, right or wrong, what you see is that, you know what, I just realized that I didn't use my little pointer thing at all. So about 23% are getting it right. 77% of them are getting it wrong. But this is not a simple multiple choice question. This is what we call an ordered multiple choice question, in the sense that the distractors here are not just any old distractors. They reflect levels of sophistication in students' reasoning that is based on research that we and others have done. So what you can see under the A, B, C pieces here, can you see my pointer? Is A is what we call a level 2, B is a level 1, C is a level 3. That's the most sophisticated level of understanding that you can have with this particular question, and D is a level nothing, because it's really mostly just a confusion here.
Ravit?
Yeah.
In order for them to see your pointer, I'm sorry, you need to hold down on it, so that way it'll show. If you just have it on the screen, we can't see it. But if you actually if you right-click and hold down as you're moving, I'm sorry, left-click and hold down as you're moving around, we can see it then.
Okay. So now you can see it? Sort of? No?
No, not sure why we're not seeing it.
I am holding
There you
them
go.
up.
Now I see it.
Okay, maybe I didn't press the mouse on hard enough. All right. So A is a level 2, B is a level 1, C is our level 3. That's the most sophisticated levels. Now what you're going to see is what students chose. So a lot of you said they were going to choose level 1, and you are in fact correct. Most of the kids are choosing a level 1. There are quite a few, though, that are reasoning at a level 2, which means that they understand that the genetic information codes for proteins. That's this. And there's some, about, I don't know what, 22% of them that are actually picking a level 3, meaning they understand that these are instructions for assembling proteins from amino acids. What this means, if I interpret it, is that most of these high school kids are really reasoning at a late elementary to middle school level, because as I showed you before in Appendix E, late elementary is understanding that genes have information for traits, and middle school is understanding that they're coding for proteins. So most of these students are reasoning at a late elementary, middle school level. So the question is why? What's so complicated about this? Well, here's where it gets really challenging for kids and somewhat counterintuitive. If you think about the biological system, right, we have multiple levels of organization with protein, cells, tissues, organs, and so on. The question is, where does the genetic information, where does it interact with all these physical? Does it code for the whole person? Does it have instructions for how fast your heart should beat? Does it have instructions for what your cell should be doing? Where is this information sort of impinging or mapping onto the physical system? And there's been research that shows that the kids have a hard time with that. So initially, when they think about genes and what genes do, they think about them as passive particles. Genes are just associated with traits. These kids don't even realize that genes are informational, okay? This is really just synonymous, genes and traits, and they don't really have a mechanism for understanding it. As they develop in their understanding, they start thinking about genes as instructions, okay? Genes have information about you. But here, these kids are really thinking about information at all levels. So you'll hear things like, yes, if you have a genetic disorder that is irregular heartbeat, it's because you have a gene that tells your heart not to beat regularly. or you have a gene that tells your, how tall you should be or what color your eyes should be. So it's non-specific information. Moving on from that, a more sophisticated understanding is what we call genes as productive instructions, and this is instructions for proteins. And this is really counterintuitive. If you think about how complicated a human being is, to think that the genetic information only codes for proteins, the only thing this information says or tells is how proteins should be made, that is, that is quite fantastic in some way, that everything else that you see about you, it really emerges from the interactions and the functions of these little machines that are called proteins. So, it takes a while for kids to really wrap their heads around this. A corollary to this understanding is that in order to really understand the gene-trait connection, you need to have quite a robust understanding of proteins and their biological role. You need to understand what proteins do, why they're important. And it's a little sad, but we don't necessarily do a very good job on that. So proteins are kind of the missing level in instruction. And we explain the central dogma. This is high school, right? We explain the central dogma in relative detail. If you open most textbooks, a lot of information there. But then we kind of drop the ball in terms of what proteins should be doing. So let's do another quick little poll in terms of what students know about proteins. How do you think a typical high school student would answer the following item? Janice and Bill are arguing about what proteins do. I'm going to let you read this. I already told you B is correct, okay? But I want you to think about what would a high school student answer. So take a couple of seconds to do this poll, and then we'll see what you say, and I'll show you what we found.
Okay, polls should be open. I'm going to look to make sure that, yep, the polls are open. You will select A if you believe Janice is correct, B if you believe Bill is correct, C, Janice is mostly correct except that she left out that proteins give us energy, and D, Bill is mostly correct except that proteins do not give signals to cells. And again, I'm from the Chicago area where we vote early, vote often. It'd be great to try to get as much participation in this as we can. So go up to underneath your name there, mouse over the A, and make your selection. And Ravit, as soon as you're ready, just let me know, and I'll go ahead and lock the polls for you.
Okay, I'm going to give folks a couple more seconds to make your selections. Vote with confidence. And why don't we close it?
Okay. Lock the responses, and I'll publish these to the whiteboard.
So we have quite a spread here. A, B, C, both A and B. Janice is correct. C, the idea that, well, C is Janice is mostly correct except for the energy. And so let me show you what happens when you give this to children. And what you find is that, again, if we just look at this as a regular multiple choice, 34 about correct, 66 get this wrong. And here's what they're actually doing. Okay, so I mapped out the levels again for you. Janice is correct as a level 1. Bill is correct as a level 3, the most sophisticated level that we have. We've decided to go with C as being a level 0. It's not so much that it's wrong entirely, but this notion that proteins give us energy is very pervasive and not really the idea, the understanding that we want kids to have about proteins. And Bill is mostly correct, except proteins do not give signals. That's a level 2. And what you see here is that a lot of kids are at a level 0. This notion of proteins is giving you energy, very, very common. Some of them also think level 2 and 3. So we have students that think at a level 3 that Bill is correct and the proteins do all these things, but we also have a lot of kids at a level 2, which is the idea that they do a lot of these things, but they don't give you they don't signal the cells. So a few key points to sort of summarize this. Viewing genes as productive instructions for proteins is a big conceptual jump. It doesn't come easy. You need to spend time on task with that. It's really important to build up students' conceptual toolkit of protein functions. And it's very important to help kids generalize. So you can give kids a lot of phenomena, all sorts of interesting disorders, and have them think about them. But if you don't help them generalize, they're not necessarily going to generalize themselves. And the generalized schema that I'm talking about here is this idea that normally a gene codes for a protein. Protein has some particular function, and then you get a trait. If there's a mutation or a change in this recipe, you get a non-functional protein, and you get an altered trait. Having kids understand that this is a very generative idea that will help them reason about a lot of phenomena. So now we're going to shift gears again a little bit and think about conceptual obstacles for classical genetics. And I've added some of our favorite little models. These are middle school kids drawing these pictures. We asked them to explain how traits are inherited, and these are the kinds of models you get. So kids in elementary have what we call a theory of kinship. So they do actually understand that offspring look like their parents. They understand that a kitten is going to look like a cat, and it's going to look like the cat that's its mother and father, not like some other completely different looking cat. But it's not mechanistic, so they don't really understand why it looks like the cat, like its parents. They do not always assume equal contribution by parents. If you look at this little drawing here and you read, it kind of said half and half, and then I don't know why this child crossed it out, but then it said a third and 2/3. This is actually quite common. The mother who's pregnant, because the baby develops in the womb, there's this assumption that the mother contributes more. There's also an assumption oftentimes that mothers contribute more to girls and fathers contribute more to boys, because for the most part, you know, girls look like their mothers and boys look like their fathers in some of, you know, some core physical attributes. So, you know, it's reasonable where these come from. The idea of two alleles is not obvious. I've already mentioned this notion of skipping generations. A lot of kids out there that are quite happy with genes completely skipping generations, meaning that there is no physical continuity to the movement of information from one generation to the next. When they see skip, they mean skip, it's gone. The parents do not have any of it. And work by many folks, but including Stewart and some British researchers, Lewis and Wood Robinson, has shown that, you know, students can oftentimes use these representations that we give them, like Punnett squares, not really understand what they're doing. So this little diagram here shows that quite clearly. We have a Punnett square. We have some notion of dominant and recessive, but it's really odd, and I don't quite know, you know, how to make sense of what this student is doing, and it's not clear that the student fully understands what they're doing. So this is something that we need to be aware of, that sometimes students can look like they're understanding when they're using these representations and not really understand it. And one of the questions that came up earlier is, how should we be teaching meiosis in the middle school? Well, the answer to that is really input-output. You have a whole set, and then you end up with half a set, and let's focus, if at all, on the steps. And we'll get back to that in a little bit. Here's talking about meiosis, here's another typical question. We're not going to do a poll on this one in the interest of time. We're just going to sort of talk through it, and I want you to think through it with me. So how do you think a typical high school student would answer the following item? Here's the item. A human male has the following chromosome pairs in his body: an X and a Y, 3-3, and 4-4. Which chromosome combinations will be in his sperm cell? And I have circled the correct answer for you. Here, essentially, we're splitting the X and the Y. We're basically splitting all the pairs, right? We're splitting the XY pair, we're splitting the 3 pair, and we're splitting the 4 pair. This is a case in which we split some pairs, but not others. We split the X, but then we didn't split the 3, and the 4 is gone because we cut it. You know, this is the shoe analogy where you get two stilettos and no boots, or 2 boots and no stilettos. This is another version of splitting, but again, they didn't split the X and the Y. So they split the 3 and the 4, but the X and the Y really trick kids up. They don't quite know how to handle that because it's not exactly the same. And here they're not really splitting correctly either, right? So this is not half. It's you have 6, half would be 3, not 2. So what do kids do? About 31% of them get this right, which means 31% of them selected A. A bunch of them are selecting C and B. So they're splitting the stilettos and the boots not correctly, or they're not splitting some pairs and splitting others. Not that many kids are actually selecting this, which is great because, as I told you, they learned genetics already. And there's some that are selecting, as I said, that are selecting a level 3. But most of them are level 1, and we said that the way we've sort of analyzed this item is that B and C are both level 1 understanding. So this is tricky for kids. So a few key points about the classical genetics bit. Focusing on details of processes like meiosis does not necessarily lead to understanding the big idea. Just like focusing on the central dogma details and the A site and the P site and tRNA and mRNA and RNA does not actually lead them to understand what proteins do and why they're important. Similarly, we can have students using Punnett squares and those kinds of representations, not really understanding what they're doing. And I think a key point here is that you really want to build kids' ideas first and then label them second. So we want to push for mechanism here and then label it. In fact, one of the biggest problems in genetics is that we have so many terms and so many different representations. Think about how many different ways you know to draw a chromosome. It is really confusing for kids. So it's important to help them build the understandings and then put the labels rather than the other way around. So we're going to take a little break for questions and then see also how we're doing on time.
Great. Yeah, I think we're doing well here.
Yeah.
And so we're going to go through a little bit and look for some questions. Please feel free, folks, as you people have been sharing some great resources they've known about in the chat room and things. Feel free, folks, to put in some some of your questions that you've got for Ravit in the chat, and we'll put those on. I had a question and I just literally forgot what the question was. So now I'm it's classic drawing a blank. Just jumping back a second here to look and see if I can find what it is. I'll also throw one out that I had earlier. You talked about the different models, and I'm probably going to pronounce it wrong. I understood most of the different models you were mentioning, except the meiotic. I think that is what you said, model. And can you refresh at least my memory on what that model is versus the others?
Yes. So basically, that model helps explain how you get to half, right? So if your parent cells, by their nature, have the pairs, they have the complete set because otherwise they would not be alive. You need to have some sort of mechanism to generate half a set, and the half a set only exists in the sperm and egg cells. All the other cells have the full set, and the sperm and the egg cells have half a set. So in the middle school, we think about it as input-output. You have pairs, and you're splitting them, cutting the pairs in half. Each pair gets split. And in the high school, we deepen that to understand that sometimes those pairs even swap parts and then split. And there's no real need to focus on the specific steps of the process. It doesn't actually help kids understand what's going on. The problem here, this is actually an evolutionary problem, right? So meiosis capitalizes on the cell's machinery to divide, which is the process of mitosis. And in doing so, instead of splitting in half, which would be the sensible thing to do if you were to design the system, they duplicate, split in half, and split in half again. So for every original cell that you had, you actually get 4 sex cells. There's no real reason for that because, you know, if you were designing the system, you just get 2. You just split it in half. But this is not a design system, as we know. It evolved, and it evolved to capitalize on existing machinery. So that's why understanding the details of meiosis doesn't actually really help because it's sort of a bit of a backwards kind of system.
Great. We've got a question here from Joan who asks, do you see NGSS supporting the topic of epigenetics?
Yeah. So I think so. I see the epigenetics piece coming in is really with the environment, and there is a huge emphasis on the role of the environment. And when I said environment, if you remember, there was also gene-to-gene interaction. So that's where I see epigenetics coming in. And you know that the word itself doesn't appear there, but the notion of environment impacting gene expression and interactions between genes is sort of, it is there. But definitely the environment impacting gene expression is, so, you know, which is one aspect of epigenetics. There's also, epistasis is not as much in there. I didn't really see it. That is quite an advanced concept, and not everyone is teaching it even now with the existing standards. But I do think that there's room to teach it. You also have to remember, the NGSS are sort of the lowest common denominator. No one is saying you can't go beyond it, but you want to go beyond it in productive ways. You want, if you're going to go beyond it, teach ideas that have power, that will allow students to reason about an even wider array of ideas, rather than just adding details that are not really generous.
Great, thank you. So why don't we go on to the next piece here, and we'll just try to do some questions at the end.
Okay, great. So I wanted to have one slide that kind of tries to do a little bit of contrast between the NSCS and the NGSS, and this is what I sort of see as major shifts. One is earlier and more extensive focus on the mechanisms that link genes to traits, right? The proteins, it's early and often, just like in Chicago. Greater stress on variation and its sources, so not just variations from independent assortment and segregation independent assortment, not just mutations, but also variation due to environmental interactions and recombination. And emphasis on the interplay between genes and the environment. That is really, again, a sort of an early and often feature of the NGSS that I don't think is quite as emphasized in the NSCS. Now that I think about teaching strategies and resources, so I would be completely remiss if I did not mention inquiry practices here. We want to be teaching these ideas by engaging kids with the inquiry practices, okay? So they're actually learning the genetic DCI through engagement with these practices. And if you look at the standards, so these are actual standards, the blue font here are the pieces of practices that showed you that earlier as well. So here for the little kids, the younger kids, we have make observations to construct an evidence-based account. Third grade, we have analyze and interpret data to provide evidence that plants and animals have traits that are inherited. And for the older kids, we have develop and use a model to describe why structural changes to genes, mutations, may affect proteins, and so on and so forth. So this is lovely. What do you actually do in the classroom? So here are some examples. If we're working with the younger students and we're talking about making observations to construct an evidence-based account, essentially the evidence here are the observations, right? So, I mean, it'd be great if you could bring animals into the classroom or other organisms, plants, whatever, to help them see similarities and differences. I happen to find 2 pictures on mine, and you can very clearly see that there's some similarities at the big ears and so on, but the color is different. Color is different between the puppies, and the color is different between their puppies and their mom or dad. I'm assuming that's the mom. Same thing for the kitten. Color of the eyes are different. Color of the fur pattern's a little different, and so on and so forth. So, you know, I think the eyes actually change. But never mind, it's still differences that little kids can notice and talk about and compare and identify ones that are inherited versus ones that may not be inherited. So if the mother cat had her tail chopped off, or it's actually more with dogs, the puppies will still have a tail. So you can think about those kinds of comparisons. With the older grades, when we think about 3rd grade to 5th grade, here we have analyze and interpret data to provide evidence. So here we want this evidence to be more generated by the kids and using sort of more tools, right? So in 3rd grade, we want to start introducing more of the scientific tools with which we can reason and quantify, measure, capture variation. One really neat way of doing that is Wisconsin Fast Plans. Okay, so if you use Wisconsin Fast Plans, you can breed them very fast, and you can make some observations that don't really require measurements and other observations that do. You can compare plants to their sort of parents and plants to other plants that are not their parents and analyze. It's essentially very similar to what you're doing with the 1st grade, but you're deepening the notion of evidence and how you get this evidence and what is not inherited. If we think about older kids, the middle schoolers, right, develop and use a model to describe why asexual reproduction results in offspring with identical genetic information, sexual reproduction with genetic variation. So this is actually from materials that we've developed and used with middle school students. What we have students do is develop a model of the rules of inheritance. So how do they think traits are inherited? We don't teach them anything first. This is essentially the first thing they do in the unit. We give them evidence in the form of either, this little table that shows Sarah, a particular child, and what her father has and what her mother has, and or pedigrees, and very simple pedigrees. And what the kids do is they work in pairs or in groups, and they develop rules. So if you look at the first rule, this is a rule after they've seen just a little bit of evidence. You get mostly observational types of things. Both parents don't have to have the disease. We're talking about albinism here. Disease can skip generations. At least one child will have it. These are all observations. There's really no mechanism here to explain why this is the case. With additional evidence, though, the kids revise their rules. And you can think of these rules as a model, okay? This is a model. And here they say, well, we have 2 genes. So they essentially invented this notion of 2 genes to explain the phenomena. Genes and traits can skip generations. And then this notion of overpowering and dominance that they have, again, sort of invented for themselves with the evidence that we provided. And using this, the teacher can then build on this and essentially do what we call a benchmark, which is teaching, Mendel's stories and Mendel's laws. But they can actually uncover a lot of this idea, a lot of these core ideas by themselves with appropriate scaffolding. Oops, what happened here? For some reason, my slide is awfully small. Is everybody seeing the slide really small, or is it just me?
I think it's just you.
Okay. Okay, I don't mind if it's just me because I know what's on the slide. Okay, so you can also do this with older students. In middle school, you can do this with in high school, but you can also do this for molecular genetics. Okay, so this is another model that kids develop based on evidence that we gave them. In this case, the evidence was related to how genes bring about the traits, right? So this is the protein story. And we did it sort of like a puzzle in pieces where they would get evidence about different organization levels. So what do the cells of normal individuals look like compared to the cell of people with sickle cell anemia? In this case, it's blood cells. What do the proteins of people with sickle cell anemia look like compared to the proteins of normal people? And they developed a model to explain sickle cell from the gene, and you can see that this child sort of has the idea of coding to the protein, to the cell, to the tissue, which is essentially the blood, and then the organism, which were the symptoms of this particular disorder. So other resources. These are several of my favorites, but there are a lot of others, and I kind of was noticing in the chat that people brought up a few really good other ones as well, so I'm glad the chat's getting recorded. Concord Consortium brings to you all sorts of wonderful things, and I think that some people have mentioned Molecular Workbench and Geniverse. There's also Geniville, which is a little different, but all of these are free. And they're really groovy. The Geniverse is actually a game environment with challenges that involves breeding what are called drakes. These are sort of small versions of dragons. And it's very engaging and quite effective. AAAS project, which it is very familiar with, has wonderful assessment items. And the nice thing is that they have kind of assessment items like what I showed you, where they tell you what are common responses for different age groups for that item. So they're both good ideas for nice items that you can add to your assessments and to help you get a better handle on where the students are reasoning, at what level they're reasoning. And the last resource is WISE. WISE comes to you from Berkeley. The latest version of WISE has I don't even know how many different learning environments and sort of curriculum materials, but there are also some in genetics. And one of the researchers that works on that is Michelle Williams, and she has units that are on WISE for 5th graders using Wisconsin FAST plans. So those of you that are teaching elementary and are wondering, oh, what do I do about the standard? That's a really good resource right there. The Berkeley WISE, Michelle Williams is the investigator, and they use Wisconsin FAST plans, which are just all around wonderful. So that pretty much brings us to the end of the presentation that I prepared. I also want to thank my former student, Nicole Shea, who helped work on a chapter that sort of formed the basis for this presentation. My email is there, so if you have questions that pop up later today or in the next, I don't know, couple of weeks, just email me and I'll do my best to address them. And thank you for being so patient and attentive.
Thanks very much, Ravit. Lots of really good things. I've learned some pieces tonight and wanted to do. We'll be here to do a couple little additional questions here at the end of the evening, but for now I want to just point out a few other places you can find information that you want. Obviously the standards themselves can be found at nextgenscience.org, and a lot of great resources that NSTA has assembled together. I love them. NSTA.org/NGSS is the home of the NGSS at NSTA hub. Point out if you want to continue this discussion, we've got a discussion forum on the NGSS in the Learning Center, and also a member-only listserv about NGSS. Both of those are great places to gain some guidance. Want to point out we've got some upcoming webseminars. We've got last one in this biology series, or life science series, Biological Evolution, Unity, and Diversity, coming up in 2 weeks. We've done bunches of web seminars covering the practices, the cross-cutting concepts, the core ideas, assessment. All of these are here and freely available to go back and look at. And a number of our articles in our various journals have been put in place here as well to use and try to go through. Our virtual conference just coming up here March 8th. Really, I'm so excited about this. A lot of folks, Ravit knows some of the speakers who are going to be here as well for this piece, really looking at the practices. And so would strongly recommend you check that out. Obviously, the various books we've talked about, the framework and the standards, and NST has a reader's guide to both the framework and the standards. Also, Roger Bybee did a book called Translating the NGSS for Classroom Instruction, and uses a lot of biology examples in that because that's his area of expertise. He was the team leader for the writing team in life sciences. And finally, as always, there's an app for that. Go ahead and look for the NGSS app, which also has some links to some NGSS resources from NSTA. I want to point out our conference coming up in Boston. I hope that some of you will be there. Please look for us at the NSTA Expo area, the NSTA booth. I also want to point out while mentioning this, also our STEM forum is coming up in New Orleans in a few weeks. We've got a link there also in the chat room. And with that, just another thanks to Ravit, and thank you all of you for being here and being involved. And with that, I'll pass things to Sue to carry out the next piece.
Thank you, Ted. And thank you, Ted, for your part of the presentation tonight, and to Ravit also for your presentation this evening. And to show our appreciation, let's go to the emoticon, the smiley face, and let's give them a round of applause for a phenomenal presentation. Very, very interesting, very important to my own presentation, or my own teaching skills, actually. And then also let's please remember to give a round of applause to Jeff. He does a lot of work behind the scenes that we don't necessarily see getting done, but because of him we're all able to stay connected. So thank you, Ted, thank you, Ravit, thank you, Jeff. And then I'd like to also say thank you to the Carnegie Corporation in New York for sponsoring today's program. And thank you to the administration of NSTA for their support of web seminars. You can find resources related to today's presentation by going to the web seminar collection in the NSTA Learning Center, which will include the archive of the web seminar and the presentation slides. The collection may also include journal articles and lesson plans, interactive content modules called Science Objects and SciPacks, and links to helpful websites and multimedia. I'm going to put a direct link to the collection in the chat window. Give me just a second to scroll up to it. I want to make sure I just get the collection. I'm sorry about that. I'm going to post that direct link into the chat window. And also, everyone who registered for the program will receive an email within the next day or so with a link to access the archive of this presentation and a link to the collection. Okay, here's a look at what's coming up in the web seminar calendar. On February 26th, we have Engineering Design Challenge, Spacecraft Structures, which features an engineering design challenge for teachers of students in grades 5 through 9. On February 27th, we have Polar Science and Engineering, a model for NGSS practices, where presenters will describe one of the latest polar research mysteries for teachers of students from grades 8 through 12. And then Skeletal Systems of Human Physiology in Space on March 4th, where teachers of students with grades 9 through 12 will learn the effects of microgravity and countermeasures that NASA uses. You can register for upcoming programs for these web seminars by visiting learningscenter.nsta.org/webseminars. Thank you everyone tonight for your participation. We look forward to seeing you again on another NSTA web seminar soon.